Hi, my name is Paul Demers. I'm the director of the Occupational Cancer Research Center here in Toronto, and I'm also a professor at the University of Toronto and the University of British Columbia in Vancouver. I'm an epidemiologist and have participated on a number of different IARC committees in the past, and in particular for the example that we use as a case study, which is shift work involving disruption of circadian rhythms. I chaired the epidemiology committee. The objectives of this workshop are listed above. The major objective is to, give people a much better understanding of the process that's used by IARC to classify carcinogens. And one of the major issues in this is also how IARC systematically uses data from a number of different scientific areas of expertise when it goes about classifying a carcinogen. And so we'll try to give you a sense for how that actually works. IARC's classification of carcinogens really plays a very key role in cancer prevention in terms of setting policies and regulations, and an underlying goal of this is to develop a better understanding of that so that we know better how these classifications can and should be used. What we'll be doing is running through a case study of how shift work was classified as a probable human carcinogen by IARC back in 2007. So IARC, the International Agency for Research on Cancer, is actually a specialized agency of the World Health Organization and therefore part of the United Nations. But it's run a bit differently, and it has a different governance structure than the rest of the World Health Organization. One reason is that it's not supported by WHO funds. It's supported directly by contributions primarily from the most industrialized countries in the world. They contribute to the overall budget, the underlying budget of IARC, although the Monograph program itself gets a bunch of separate funding as well that I'll mention in just a minute. And that means IARC has different hiring policies than the WHO and a number of other differences. Also, it's not located in Geneva. It's actually located in France, in Lyon, France. Now, IARC has, at this point, reviewed about 900 agents for their carcinogenicity since the program was started in 1971. We use the word agents because IARC actually looks at a number of different types of things that are suspected of causing cancer. Now, sometimes that might be chemicals, it might be a mixture. It could be physical and biological agents. So it might be, for instance, a particular virus. It could be different forms of radiation, or both ionizing and non-ionizing radiation. They really have a very broad mandate. And it also includes things that we call exposure circumstances, which is a little bit more difficult to get a handle on, but certainly shift work qualifies as an exposure circumstance. It's not kind of a measurable exposure per se. It's a situation that results, or at least is thought to result in an increased risk of cancer. The way that IARC chooses its priorities for what it's going to review is that about every 5 years or so it convenes a scientific committee that makes recommendations on what the priorities should be for the following 5 years. But on top of that, it also takes recommendations from the other member countries that contribute to the, to the budget of IARC in terms of supplying funding, and they also try to coordinate with organizations such as the U.S. National Toxicology Program. But there is a kind of a high-level discussion that happens between a number of these regulatory agencies and IARC when it comes to setting their priorities. Now, an actual evaluation that's done by IARC is done by what's called a working group. It's a group of 20 to 30 international scientists. They're people who are recognized for their international expertise. They're also people who are selected because, or at least initially because there are no known conflicts of interest. Now, members of the working group are initially filed into 4 different subgroups that I've listed here. The first one deals with exposure data. The next group is the studies of cancer in humans, and this is basically a group of epidemiologists. The next group is studies of cancer in experimental animals. The last group is called mechanisms and other related effects, who review the data regarding theories about how something might cause cancer, so mechanisms of cancer. So what you have here in a particular committee is a very interdisciplinary group representing a lot of different branches, a lot of different types of scientists. So a large body of literature is kind of made available to you, and you have 6 months to review it and to produce a first draft and to send it to the rest of the working group in time for them to read it. The actual evaluation, the formal evaluation, takes place during an 8-day period. The initial part of the meeting is actually the first days people are in subgroups, so we break into those 4 different groups. In the last days of the meeting, you go into the mode where the entire working group is there, and it's a fairly formal process leading to eventually a series of votes that lead to the classification. And then shortly after, generally within days of the meeting, but certainly I think within a week, what you'll see is coming out of a meeting is something that looks like this. And this is, IARC produces kind of a summary of the monograph that they actually produce for Lancet Oncology. The full monograph, this kind of paper thing that you find on library shelves, used to take approximately a year, now takes, is generally backlogged several years. The process, I've kind of just laid out here a real simple diagram to talk about the process that IARC uses. What you see here is, on the top row is cancer in humans and cancer in experimental animals. Those 2 committees, in reviewing the set of literature that they have, use the rules that are described by IARC. They're certainly presented to us multiple times during the IARC meeting. You also see them summarized in what's called the preamble. That's kind of the working, it's a little bit of background information, but it's also the working procedures of an IARC monograph group. So the preliminary thing is that each of those groups comes out with an evaluation. We come up with a preliminary default evaluation. We try to consider other relevant data and then come at a final evaluation. Now, the final evaluation that people end up in is one of these 5 groups that I have laid out here. I mean, group 1 is a definite human carcinogen, fairly high set of evidence to be met for that. Group 2A, that it's probably carcinogenic. Group 2B, it's possibly carcinogenic. Group 3, it's not classifiable. And this, it's important to recognize that this is the neutral category. Not classifiable means that we simply don't have sufficient evidence available to make an evaluation and actually say whether something is even possibly carcinogenic. And group 4, evidence suggesting a lack of carcinogenicity. There are 900 different things that have been evaluated by IARC. This is from the IARC website, and this is how it parses out. Many things end up in this not classifiable category, actually over half of them, which in some ways is disappointing, but it really reflects how much data is available.